Hey all, we today are going to be looking at HL content C1.1 for metabolism, and we're going to be looking at the interdependent components of metabolism. And so our theme here is interaction and interdependence, the interaction related to enzymes, and now the interdependence of how is metabolism connected to those enzymes. So when we're thinking about metabolic reactions, we need to recognize that metabolic reactions produce waste heat, which organisms use to maintain their body temperatures. Sometimes excess heat is generated, for example, by exercising, and then it's gotten rid of by sweating and evaporative cooling, dependent on how an organism might utilize structures in its bodies to evaporatively cool. Mammals and birds can raise their body temperatures using muscle contractions. So obviously that would be exercising because we are using up energy in our bodies, and that's going to produce waste heat. And additionally, using something called brown fat, which is a type of fat cell that contains lots of mitochondria, but those mitochondria are decoupled from respiration. So they don't make ATP, but they do make heat. And organisms that hibernate, for example, like bears, have a high percentage of brown fat in their bodies. Intra and extracellular enzymes is the next slide here, and we're going to be thinking about enzymes being synthesized by ribosomes. Enzymes are proteins, so they're going to be made by ribosomes. Exoenzymes are released and work outside of the cell. They're created by the ribosomes, sent to the endoplasmic reticulum, and then to the Golgi body and move out. So enzymes, for example, in digestion, those would be functioning in this way. In unicellular organisms, exoenzymes are released to digest macromolecules into monomers that can bypass cell walls into the plasma membrane. So they're actually releasing enzymes externally to their own bodies. Intracellular enzymes are made by free ribosomes, so they're not attached to endoplasmic reticulum, and they're going to be used for processes within the cell. So examples of reactions that would require intracellular enzymes would be things like glycolysis or the citric acid cycle, which is also sometimes called the Krebs cycle. So enzymes can be inhibited in different ways. The two ways that we're going to talk about here are competitive inhibition and non-competitive inhibition. Metabolism is typically occurring in a sequence of small steps, and those are catalyzed by many different enzymes. And there's a bunch of branches that are going to occur in a metabolic pathway. So here, this image is showing a bunch of the different branches that could occur in a metabolic pathway. That branching is quite common. So competitive inhibition, a molecule is going to compete for the active site location with the substrate. And so once the inhibitor is in the active site, the reaction is going to stop because the substrate can't get into that active site. And the inhibitor is going to stay in the active site until the concentration of the substrate decreases sufficiently that the inhibitor will detach from the active site. In non-competitive inhibition, an inhibitor is going to bind onto a secondary site called the allosteric site, and it's going to change the shape of the active site. So it's going to prevent the substrate from getting in there. So it's not competing with the substrate, but it's forcing the situation such that the substrate cannot bind. So there's naturally occurring non-competitive inhibition occurring in our bodies, but this can also occur as a byproduct of chemical interactions. So in some cases, chemicals can bind into the allosteric site irreversibly, which then means that enzyme becomes inactive so that it can't work with the substrate and the inhibitor can't detach. And then that means that that enzyme will just no longer function. So when we're thinking of inhibitors, you can see on this graph here, as the substrate concentration is increasing, the rate of reaction is also going to increase just because of more bonking together. If there's no inhibitor, eventually that's going to plateau. If there's a competitive inhibitor, so it's binding onto the active site, then eventually, if we increase the substrate concentration more and more and more and more, there's going to be more and more and more collisions. And eventually, if the substrate concentration is high enough, even with a competitive inhibitor present, then we're almost going to get to the same kind of reaction rate we would see with no inhibitor. This is not true of non-competitive inhibition, because no matter how much concentration we add of substrate, the inhibitor has still bound onto the allosteric site. And so that means that the active site, it doesn't matter if the substrate is colliding with the active site, because the active site has changed its shape so that that substrate can no longer bind with it. And then also what we're looking at here for different kinds of chemical reactions, if we're looking at a chain of reactions here, where we have something, the product of a chain of reaction reacts with the allosteric site. So you can see here, there's the allosteric site right there. And it's going to inhibit the chain from proceeding. So once there's enough of this end product, this is called feedback inhibition, the end product is going to actually bind onto the allosteric site, change the shape of the active site, and then the pathway shuts down. And it's going to interact with enzyme number 1, because it doesn't need to go through all of these steps in order to inhibit. It wants to limit the amount of energy that's going to be spent on this chemical reaction pathway. And so once there's enough of it, it's going to attach onto the allosteric site. And then similarly, once there's too little of it, then it's going to detach from the allosteric site and reactivate this enzyme pathway. So certain heavy metals, for example, are irreversibly maybe going to attach to an allosteric site. So an example would be mercury. It's going to bind to the active site and it's going to make them not work. So this is a mechanism-based inhibition, and it can have very serious health consequences, which is why we don't expose ourselves to mercury. So to summarize, enzymes catalyze reactions by lowering activation energy. We have intracellular and extracellular enzymes. Competitive inhibition is where the substrate is competing with another molecule for the active site. This is mechanism-based. Non-competitive inhibition, or it can be mechanism-based, I apologize. So mechanism-based would be where a chemical such as mercury is binding into the active site and cannot reversibly remove itself. Non-competitive inhibition is where another molecule is going to bind to the allosteric site and it's going to inactivate the enzyme by changing the shape of the active site. And then feedback inhibition is where that final molecule in a chain of reactions binds to the allosteric site to deactivate the initial enzyme in the pathway, preventing that pathway from continuing for a time. Okay, that's it for this video. Thanks for watching.